Hello to my online learners. Well, it's time to do one of my least favorite organs, the brain. The reason the brain is my least favorite organ is that it's not clear. It's not this part does this. There's sort of plasticity, which means that one part can take over for another part. Plus, when you look at a brain, and I guess I've seen preserved whole brains, there's not nice little lines in it. It's kind of all together. You can see different parts, but not clearly. So what we're going to do with this chapter, and unfortunately because there's so much material and so little time, this is basically name that part, what does it do? The brain is fascinating, very, very fascinating. I took a couple courses on it and it fascinates me but frustrates me at the same time. The unique thing about the brain is that it has plasticity, which means one part can take over another function. So especially if it's a young brain. So if it's not getting proper blood supply, it can reroute the synapses. So it's fascinating. And this is why after a stroke, with time, there can be some recovery. Or if there's brain damage as a child, with time, there can be some recovery. Now, as I say, with this chapter, there's a lot of name that part, what does it do? So here is the reading guide, and on 22, we're going to work on through 22, 23, and 24, which is basically name those parts. Now, to help summarize, I'm going to open a Word document and make a summary table because that's what I like to work. Anyway, I'm going to move this over to the other side here, and then I can see it. Okay, so to create a summary table, we're just going to go File, Insert Table, and drag down. I'm not sure how many, but we can always lose or gain from there. Start with a title, the brain. Make sure you save it so when my system crashes, I have it already, which is cool. And there we go, it's saved. Good. So let's put in just start by putting in the topics and then we'll fill it in later. So brainstem is, now, first off, my brother's name is Brian. And all my life, I have mixed up Brian and brain. So if I put in Brian instead of brain, that's because of my brother, okay? So warn you in advance, I do know the difference between a brain and my brother. However, my hands will type a little thing different. So brain, cerebellum, cerebellum. Then we have the diencephalon. So there's two parts in that one because there's a di. Then we have the cerebrum. And then we have the limbic system. That one's usually, most people aren't aware of it. Come on, fingers. Limbic. Forgot the B in limbic. Limbic system. And then we're going to put meninges in here. Meninges and cerebral spinal fluid, which we're going to abbreviate CSF. And if you put the abbreviation in here, then we can use it later. Now, we can fill this in as we go. Oh, sorry, I forgot a title. So let's put a column on top. So this is our structure. And then we're going to have our description because it says we need to know the major landmarks. So basically, what is it? And then our function. Now, from our definition here, we need to know the landmarks, the subdivisions, and their functions. So that's what we're looking at. So here's our summary table. We'll fill in as it goes. Of course, a summary table is not perfect. It's merely there to help organize data. If you don't like a summary table, do a flow chart or whatever you wish. I just find when I'm typing, it's easier to put it in a table and then I can jump around. Okay, let's go back to the textbook and see what the textbook says. And we'll work through the textbook. That's not the textbook. Come on. Okay, to the textbook. Here is our textbook. Let me move that down. Now, this is starting on page, I don't even know what page it is, just a second. Let me move that over so you can see. There we go. So, we are starting on page, I don't know what page, page with a brain that looks like this. So what we're going to start with, this starts, so generally we have there's the cerebellum down at the bottom, which they say looks like a cauliflower. You've got the brainstem. The brainstem is made up of the midbrain, pons, and medulla. So let's add that to it so we know what we're looking for. Let me change the setup a little so it's working better. I wanted to do this with, there we go. So in the brain stem, we've got the midbrain, pons, and medulla. Just so we're aware of that. And since they are parts, I'm going to indent them. And indent. So this way we know they're subdivisions. Okay, good. So that we can get that from that. Then we have the cerebellum is the largest region of the brain. It contains more neurons. Do we need to find that interesting? No. The diencephalon sits between the cerebrum and the midbrain. Not needed. Cerebrum is the largest portion of the brain. It has surfaces, gyri, which are ridges, sulci, which are shallow, and fissures, which are deep. Do we need to add that? Let's put it down at the bottom just because it's some nice vocabulary. And you never know, we might need a little vocabulary. So we had gyri, which are ridges on the brain. There we go. Not the brine, on the brain. Sulci, which are shallow grooves. Now, shallow sulci, that's why I use the word S. And then fissures are deep grooves. Okay, so take your time. If you need to, if I'm going too fast, stop and keep up with me. So this way, stop the video and keep up with me. So this way you can learn to summarize too as we go along, because not every teacher is this nice to me. It's too nice to you. Okay, good. So we're going to save this because I've already crashed. I've already crashed once this morning. Grrr. I'm not sure what's going on with this crazy system, but whatever. Okay. So we have, what else have we taken out here? We have 2 cerebral hemispheres. The deep groove is called the longitudinal fissure. The bundle between is called the corpus callosum. Good. Whoops. Okay. So that is pretty much that. I think we've extracted enough information off of that one. Gray matter and white matter. So, like the spinal cord, the brain contains gray and white matter. Unlike the spinal cord, which the gray matter forms inner, in the brain, the gray matter forms the surface. Specifically, the gray matter consists of cell bodies and interneurons, covers the cerebrum and the cerebellum in a layer called the cortex. Underneath the cortex is white matter, although the gray matter exists in patches called nuclei. That's not on the, interesting, but not on the learning outcomes, not needed. Meninges of the brain. Like the spinal cord, meninges cover the outer surface of the brain, offering protection. Nearby bone can help protect the bone or brain from trauma. So when we're looking at meninges, the function is protection. And then when we look at it, here are the 3 different meninges. The Dura Mater, the Arachnoid Mater, and pt1. So since there's 3 things and they're in layers, this is a good question to put in order from external to internal. We also want to know what the order or the surface is, or the order is, and the function. Okay, so the pt1 clings to the brain. The Dura Mater is the tougher one on the outside, and it's for protection. So now that we've looked at that little bit, let's put that in our summary table. So under meninges, we have Dura Mater. Is it Mater or Mater? Spelling again. With just one T. M-A-T-E-R. Dura Mater. Dura Mater. Arachnoid, and it's like arachnoid is like spider, so it would be an arachnoid space. Now, is that also a mater? No, it's, yeah, arachnoid mater and pt1 mater. So for the dura, let's put that is outer. And then down in the pia, let's put that is inner. Okay? Good. And we know that they are protection. So we've added protection there. What else does it say? Oh, they're tough coverings. Let's read again and see what else is there. They cover the brain, 3 layers. Good. What about this one up here? The sinuses collect blood and pass it to the way of the heart. We didn't mention it. The subarachnoid space is important because later on we will learn about that in some places. No, that extra part is extra. So we need to put the subarachnoid space in there just so that we know the subarachnoid space separates the arachnoid mater from the pia mater. So let's go back to our summary table, and after the arachnoid mater, let's put subarachnoid space. Okay, so this way we know them in order, and you can indent these if you want. Let's do a little indenting to show that it's a subfunction. Okay, there we go. Okay, and save because I'm crashing. Okay, so this is that. Basically, they're tough coverings. It is a form of protection. Good. Let's continue on with the next bit of the textbook. The ventricles. The ventricles are internal parts, and I think we have this thing that it's not on the list, but I want you to be aware of it, because we think the brain is solid, but there's actually a ventricle or a chamber inside the brain, and the chamber inside the brain is there for the fluid to flow through. So when we look at the cerebral spinal fluid, the cerebral spinal fluid is a clear, colorless fluid called cerebral spinal fluid that fills the ventricles and the central canal. It bathes the outside of the brain and spinal cord. It is formed from blood at the choroid plexus. That sounds interesting. And the network of blood vessels. So choroid plexus is an interesting sounding word. Let's add that. It is a clear, colorless fluid. That's important. It fills the ventricles and flows outside the brain. So let's add that. So we have cerebral spinal fluid. It is in and around the brain and spinal cord. Clear, colorless fluid. And it is for protection and bathing. The brain and spinal cord. Okay, good. Choroid plexus. Choroid plexus creates it. So let's say it's created by the choroid plexus, because that's a good term. Now, if you remember back to when we were talking about neuroglia, here's where we link back to the ependymal cells. The ependymal cells in neuroglia. I'm just going to put this in here just because. Ependymal cells. Just so this way we know that we are linking stuff together. Ependymal. Good. Save. Now, on the Blackboard page, there was a video of the cerebral spinal fluid. So let's have a look at that to see if there's more information or for you to get a little more idea on the cerebral spinal fluid. Okay, so on the Blackboard page, down near the bottom, cerebral spinal fluid here. Okay, a brief and informative look at the layers that surround and protect. And notice in this one it says, get cerebral spinal fluid, get the gist of what the fluid is and what it does for your brain and spinal cord. So we just want the gist of it.
Where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the ventricles. The ventricles are a network of cavities that are distributed throughout the brain. They are lined with a specialized membrane called the choroid plexus, which is composed of glial cells called ependymal cells. Ependymal cells are specially designed to secrete cerebrospinal fluid, which then flows through the ventricles and around the brain. Thus, the main role of the ventricles is the production and distribution of cerebrospinal fluid. Cerebrospinal fluid, or CSF as it is commonly called, is a clear, colorless liquid that performs a number of important functions in the brain. Among other things, CSF surrounds the brain, forming a protective layer and suspending the brain in fluid, which reduces the strain forces like gravity would have on the brain. Also, it constantly flows through and around the brain, removing toxins and regulating the extracellular environment of neurons.
Okay, let's stop there because there was a lot of information already we want to add to it. So it said it protects the brain and bathes it. Oh, I forget already what it says. It floats in the, suspends the brain and prevents the effects of gravity. Okay, so that's good there. Now, also, it says it bathes the brain and it flows, I guess, flows in and around the brain. So it's moving, and the fact that it's moving means that there can be problems if it gets clogged. Okay, stop that, back to the video.
There are 4 ventricles. There are 2 C-shaped lateral ventricles, one in each of the cerebral hemispheres. The Latin donut. And the section that would be the whole of the donut is an area that is wedged between the cerebellum on one side and the brainstem on the other. The fourth ventricle is shaped like a tent whose peak protrudes into the cerebellum. There are 3 openings in the fourth ventricle that allow cerebrospinal fluid to enter the subarachnoid space, a CSF-containing cavity that surrounds the brain. Thus, CSF leaves the ventricular system via the fourth ventricle. The fourth ventricle extends to and is continuous with the central canal, a CSF-filled cavity that runs the length of the spinal cord. It can result in increased pressure and expansion of the ventricles.
Okay, so there's a lot of terms there. Don't need to know terms, but notice that it flows. So if there's a blockage, that can lead to putting increased fluid, and that puts pressure on the brain. So that's a good one there. We've got that information. Let's go to this one to see if there's any more information.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the meninges. The term meninges comes from the Greek for membrane and refers to 3 membranes that surround the brain and the spinal cord: the Dura Mater, the Arachnoid Mater, and the pt1 mater. The meninges protect and provide structural support for the brain as well as contain cerebrospinal fluid. The outermost layer of the meninges is the Dura Mater, which literally means hard mother. This thick and tough layer adheres to the skull on one side and the arachnoid mater on the other side. The dura provides the brain and spinal cord with an extra protective layer, attaches the brain to the skull and the spinal cord to the vertebral column to keep them from being jostled around, and provides a system of venous drainage through which blood can leave the brain. The arachnoid mater gets...
Okay, that was really fast, but out of that we got that it also protects and suspends. Good.
Its name because it has the consistency and appearance of a cobweb. It is much less substantial than the dura. Strands of connective tissue called arachnoid trabeculi stretch between the arachnoid and pia mater. These help to suspend the brain in place. Between the arachnoid and pia mater, there is also an area known as the subarachnoid space, which is filled with cerebrospinal fluid. The pia mater is a thin layer that closely follows the contours of the brain. It forms a tight membrane around the brain and spinal cord, acting as an additional barrier and aiding in the secretion and containment of cerebrospinal fluid. Blood vessels are held against the pia mater by connective tissue before they penetrate the brain. There is a space between the dura of the spinal cord and the bone of the vertebral column known as the epidural space. Analgesics and anesthesia are sometimes administered here. Also, the dura and arachnoid mater extend several vertebrae below the end of the spinal cord, creating a cerebrospinal fluid-filled area called the lumbar cistern, where there is no spinal cord present.
Okay, again, lots of terminology. You don't need to know it, but it's good to know that the cerebrospinal fluid goes all the way around the spinal cord also, since the word spinal in cerebrospinal. And also, for those of you who've had epidurals, you get it in the epidural space. Epi means above, so it's above the durum. Interesting.
Cerebrospinal fluid can be withdrawn from here because a needle can be inserted with little risk of damaging the spinal cord. Thus, the lumbar cistern is the site where cerebrospinal fluid is aspirated in a lumbar puncture, also known as a spinal tap. This is done, for example, to diagnose meningitis, a potentially life-threatening inflammation of the meninges.
Okay, good information. Do we need anything extra? No, but now we have a visual of how the cerebrospinal fluid flows. So we know the cerebrospinal fluid flows, we know that it moves, we know that the area can be clogged, and because it gets clogged, it will back up, put extra pressure on the brain. That extra pressure on the brain can cause damage to the cells because that extra pressure is preventing a good blood supply. Okay, so that is our cerebrospinal fluid. I think we got enough information. Did we add anything extra there that we needed to know? Protection, bathing. Oh, suspension. So protection, suspends the brain, prevents the effect of gravity, holds it in place. And we have our, okay, I think we got a good summary on that. So that's enough of cerebrospinal fluid. Next topic. So if we move down, the next topic. So here's the flow of cerebrospinal fluid. Did we get everything here? Choroid Plexus produces it. It flows around. Again, more detail than you need to know, but I want you to realize that it flows around. So this way the whole brain gets suspended in it, washed by it. It flows, it flows, it flows. It flows through small openings called foramen. Then it flows back down the spinal cord. And it is, oh, here's an interesting one. It is reabsorbed into the venous blood supply by projections of the arachnoid mater into the dural sinuses. So it is reabsorbed into the blood supply. Let's put that there so that shows that it's moving. Flows around the brain and is reabsorbed into venous blood. Okay, good. Nice job. Okay, we got enough of that. Do we need a fascinating and the blood-brain barrier? No, there was no mention of the blood-brain barrier, but the brain demands high volume. We talked about astrocytes are important. Yeah, that's enough of that. Okay. Divisions of the brain. Brain stem. We'll do the brain stem. And then, so when we look at it, the brain stem consists of the midbrain, the pons, and the medulla. So let's look at, here's the cerebral spinal cord, or sorry, the spinal cord coming up. Then the green is the medulla, the pons, and then the midbrain is the blue thing. So the thalamus and the hypothalamus, that is the diencephalon. So let's look at what we've got. So the midbrain contains tracts that relay information. It contains auditory and visual reflexes, as well as controls neurons of the motor control. So it's a relay station. Let's put that in there. Midbrain. It's up to our midbrain here. Okay. Midbrain is a relay station. Good. Next, we have pons. Pons contains tracts that convey signals to and from different parts of the brain. Several cranial nerves come off of it. So, contains tracts that convey signals to and from different parts of the brain. Kind of vague, but it, relay station conveys signals. To and from the brain. Okay, good. Medulla. Medulla is attached to the brain to the spinal cord, relays sensory and motor signals between the brain and spinal cord, contains nuclei that perform vital functions to human life. So cardiac, which is heartbeat. So we've talked about the heartbeat regulator in the heart one, so maybe this is important because we're talking about it again. Vasomotor controls blood vessels and the respiratory center. So the medulla is controlling coughing, sneezing, swallowing, and vomiting. That sounds important. So medulla contains regulatory centers for basic human functions, such as heartbeat. Oh, I put them in the wrong. Why didn't you tell me? This is all function, not structure. Okay. Heartbeat, breathing. Okay, good. Now, when we talk about the description, so medulla, so on top, so the top would be the midbrain, the pons is in the middle, and the medulla connects to spinal cord. Okay, so this way you don't need to, you need to know in line what they are. You won't have to, I don't think you have to memorize, label a diagram. I don't know, I haven't finished writing the test yet. But think of it, spinal cord, medulla, pons, midbrain. LMNOP, it's medulla comes before pons, it's alphabetical. Okay, so that is the midbrain. The midbrain is often called the reptile brain. Very basic functions, breathing, heartbeat, very basic. Okay, this is already 28 minutes in, so I'm gonna stop the video here and start the rest of the parts in a second video, just so that this isn't a long video. So catch up on this if you want, get a little ahead, see how good you are at summarizing, and if not, join me for part 2 of the brain in a bit.